Hello all. So today we're going to be talking about D2.1, which is cellular reproduction. In this video, we're only going to be talking about mitosis, and we're going to be looking at how large numbers of genetically identical cells can be produced. So mitosis is the process by which your cells in your body replicate themselves. So for example, your skin cells, you need them to be constantly replicating themselves so that you don't lose all of your skin as your skin sheds. We want to be thinking about continuity and change. So in this case, we're focused more on the continuity aspect of it. How does our DNA remain consistent for each cellular replication process? The change aspect comes in when we start looking more at a process called meiosis, which is how sex cells are made. Their sex cells are called gametes. And it goes through the initial phase of mitosis with some different changes involved in there in order to make genetically separate and unique sex cells. We will learn more about meiosis later on, but this is the process of mitosis. So in mitosis, we have 4 phases. You can remember them with the acronym PMAT. So this is going to be prophase, metaphase, anaphase, and telophase. And we are going to need to know a bit of information about each of those stages. And the starting phase is interphase. So this interphase, we're going to talk a little bit more about as well. The purpose of mitosis is to replicate the DNA and make a new cell. If we are looking here at the strands of DNA, these are called chromosomes in this cell. We have one copy of our DNA from our dad and then one copy from our mom. So if we think here, these smaller chromosomes are chromosome 1, and then these bigger ones are chromosome 2. You can see there's a yellow one and a blue one. So let's say the blue ones are from dad. So this is chromosome 2, and one of them's from mom, and then one of them's from dad. And this is chromosome 1, and one's from mom and one's from dad. And so we have 2 sets of DNA, one from mom, one from dad, both chromosome 1, but one's from mom and one's from dad. So this is a diploid cell, which is going to be abbreviated as 2N, and N is referring to the genetic material. In the process of mitosis, DNA is going to become supercoiled around proteins. Those proteins are called histones in order to divide effectively. Chromosomes become visible in their X shapes at this stage. So you'll see here in interphase, it's like a giant bowl of spaghetti noodles. And in order for that separation process to happen effectively, those spaghetti noodles need to be coiled up into little packets so that they can be effectively split apart equally and evenly and properly. Sister chromatids. So what has happened here for chromosome 2, this big one, is that this was from mom and it's copied itself. That's why there's 2 strands here. And these are called sister chromatids. They are exact replicates of the same DNA. So they are held together by cohesion proteins at the kinetochore here until anaphase, when they will separate. In prophase, the nuclear membrane falls apart. Metaphase, they all line up, and spindle fibers are going to come down and attach to the kinetochore, and the spindle fibers are going to pull a segment of each of the strands of DNA apart, and then in telophase, they're going to separate into new cells. When we're thinking about mitosis, one thing that we can look at is mitotic index. And so mitotic index is measuring how many cells are in mitosis compared to the total number of cells. So in this image here, we have visible 20 chromosomes are in mitosis of a total 75 cells, because 55 of them are not going through mitosis. So we can see here the DNA is not condensed, which means that those are not going through mitosis. In these cases here, you can see they're separating. This one is clearly metaphase because it's lining up. Here, you're in prophase because the chromosomes are starting to condense, and you can see that. I don't know if we can see any telophase yet. Number 3 is a telophase, one where it is separating and the DNA is starting to uncondense, meaning it's not wrapped around its histones anymore. So if we're looking at this, 20 divided by 75 is going to give us a number of 0.267. what would it mean if the tissue, if the mitotic index was higher? It would mean that the cells were replicating faster for some reason. And so in some tissues, that's going to be possible. And then if you had a cancerous cell, that also could be going through mitosis more quickly. In the process of cytokinesis, this is how telophase occurs. Cytokinesis is different in plant and animal cells, and cytokinesis is the way to divide up the cytoplasm. So in animal cells, the plasma membrane is going to be pulled in by contracting proteins. The contracting proteins are called myosin and actin. And if you do HL and you are learning about muscular movements, then you'll learn more about myosin and actin. But those contracting proteins are going to pull in the membrane, which is then pinched apart when the cleavage furrow reaches the center. So it kind of looks like a little bum here, but the cleavage furrow reaches the center, and then there's a bunch of microfilaments that pull apart, and then you have two daughter cells formed. In plants, which is this image here, plants have bundles of microtubules that act as a scaffold for vesicles at this point here. So to make two new membranes, so the vesicles are coming here and they're joining up together to make membranes. And then pectin and cellulose are dropped off by different vesicles and through the process of exocytosis, and that's going to be dropped off between the membranes called the middle lamella to make a new cell wall. And once that's fully built, then you have a separation of the cells. Cytokinesis can occur unequally, and it does so in the case of egg cells, like human reproductive egg cells. And in order for a cell to survive, it just needs a nucleus and a mitochondria. But in the process of egg cells, what ends up happening is it's going to go through two division stages. And the first division stage, you're going to have one big cell and then a super tiny one. And the super tiny one is called a polar body. It contains genetic material, but no mitochondria. So it's just going to slowly break down. And then the second division, it's the same thing. One really big one with all of the organelles in it, and then a really tiny one with extra DNA that is not needed. So in the process of separating, as it does in metaphase and anaphase, the centromere, you saw that as like a little X black, like star shape at the tops of the cells on slide 1. The centromere is a microtubule organizing center, and it's going to contain, the centrosome is a microtubule organizing center. It contains centrioles. So that's at the ends of the cells there. These centrioles can build microtubules by creating a base structure for alpha and beta tubulin to build on. And these are proteins, so they're going to form a ring-like structure onto these centrioles, and then it's going to connect to the kinetochore proteins at the centromere. So that's the connecting point where the sister chromatids are attached to one another. And so once it's connected, so you can see here, these little strands here are the microtubules they've attached to the centromere. And then the kinetochores start disassembling these tubules, causing movement to the poles when this protein in the center dissolves. This protein holding everything together is called cohesion, and cohesin is going to dissolve, and then the kinetochore breaks down the microtubule and pulls the whole strand of DNA apart. So to summarize, mitosis is to generate genetically identical cells, and there's 4 stages: prophase, metaphase, anaphase, and telophase, followed by cytokinesis. There's also a phase called interphase, and this phase is where things are growing and then the DNA replicates itself. Most of the time, cells are in interphase. They're not going through mitosis. So about 80% of the time, cells are chilling out in interphase, just growing. The mitotic index, if it's closer to 1, shows that more cells are going through mitosis. And proteins work in cells to separate DNA through the microtubules, and that's going to cause cytokinesis. So if you're SL, you can stop here, but if you're HL, there's a bit more that you need to know. In the process of mitosis, cyclins are proteins that are going to bind to enzymes to regulate the cell cycle. So as I said before in the summary there, mitosis is only making up a small percentage of the cell cycle. Usually the cells are hanging out in G1, S, and G2 phases. And so cyclins are going to trigger these different phases. And so in G1, the cell is growing, it's creating more organelles, and the chromosomes and the cytoplasm is expanding. In S phase, the 46 chromosomes for a human are going to be duplicated. And then in G2 phase, the cell is double checking the duplicated chromosomes and making any repairs that are needed. You don't need to know what cyclin D or E or A do, but you do need to know that cyclins regulate the cell cycle. Most of the cell is in interphase. And G1S and G2 phases, we have growing organelles, cells gaining size, DNA replication happens. G0 phase is going to be a growing or differentiation phase. And so as opposed to getting ready for replication, it's going to be in a cell cycle arrest. And so that has happened in the case of spermatogonia in males or in primary oocytes in females. Until puberty is reached. Those cells are just in a resting phase. They are in G0. And so once puberty is reached and certain hormones are activated, then those cells will start going into the G1 phase. Mutations, we know of this likely as cancer, and mutations can occur to our DNA. Tumors occur because a cell replicates with damage to the genetic material, the genes, that control the cell cycle. They replicate, passing on their damaged genes. Proto-oncogenes are control genes that impact cell proliferation, growth factors, or secondary messenger systems. And so if these proto-oncogenes, control genes, are damaged in some way, then they're no longer controlling the cell cycle and they can replicate out of control. So a benign tumor are cells that are growing, but they're not spreading. Whereas a malignant tumor is the cells are cancerous, there's genetic damage that has happened, and it is spreading to other organ tissues. Tumor suppressor genes prevent cell proliferation, repair DNA, and program cell death. So there are a lot of genes in cells that are designed to prevent tumors from occurring. But if you have damage to your tumor suppressor genes also, then you can, either you have damage to proto-oncogenes or tumor suppressor genes, then that can cause cell proliferation of damaged cells. If cells detach from each other and move to different areas of the body, that is considered a malignant tumor. And chemicals like benzene, tobacco, or other like Agent Orange that was used in the Vietnam War are all chemicals that can cause damage to DNA, and all forms of high energy radiation, like from a nuclear reactor or also from the sun. So the sun can cause skin cancer, and that's exactly what's going on here. DNA is being damaged. So to summarize, usually cells are hanging out in interphase, growing stuff. Mitosis happens relatively quickly to create a new cell. So relatively quickly, meaning that the mitosis process happens over the course of 60 to 90 minutes, whereas about 24 hours is the cell cycle, where everything's hanging out in interphase. The cell cycle is controlled by cyclins. There are genetic mutations and epigenetic changes. That means changes to the genome that occur due to environmental factors, like smoking, for example. They can modify the cell cycle and eventually lead to cancer. Metastatic cancer is when cells are growing out of control and can spread from one area to another. So metastasis, if you've heard the word, your cancer has metastasized, that means it's spreading to other areas of your body and is a very serious health issue. So that is all for SL and HL for this video. Thanks for listening.